Hi, this is Gauni. It is our great honor to introduce studies on VOC using passive sampling today. While I design and manage these VOC studies, the actual work and analysis were carried out by the Mrs. Jimin Lee. Since today's presentation focuses on the technical aspect of VOC sampling, analysis, and application, I asked her to present because she has a better understanding of the details than I do. Before she starts, I would like to take a moment to briefly introduce her. Ms. Jimin Lee is a postgraduate researcher in my lab with a master's degree related to today's topic. Recently, she participated in the Asia project with Korea's NIR and NASA, where she measured the spatial variation of ammonia in Korea, the Philippines, and Thailand using NASA's DC-8 aircraft. Currently, she is responsible for the analyzing PTR mass VOC data collected by NIR's 1900D research aircraft. Please welcome Ms. Jiminy as she begins her presentation. Thank you.
Hello everyone, my name is Jinui from Hangul University of Foreign Studies. I have conducted research on measuring and analyzing real sheets using the passive sampling method. In today's lecture, I will explain the passive sampling method for real sheets based on my experiment and research. Firstly, real sheets are air pollutants that must be studied and managed as they play a key role in the formation of ozons and aerosols. There are various methods and equipment for analyzing real sheets. The equipment in this left picture is the gas chromatography that we use. This is one of the most widely used techniques for real sheet analysis. In GCs, volatile components in the samples are separated and sent to the detector, where their concentration is analyzed. GC is typically combined with a flame ionization detector or mass spectrometer to identify and quantify real sheet components. It is particularly strong in separating various real sheet components individually. Next, the equipment on the right is called a PTMS. The biggest advantage of this instrument is that it provides real-time concentrations measurements. This technical detects real sheets by transforming protons to ionize the sample gas. Since it uses protons transfer reactions to ionize volatile compounds non-destructively, so it provides real-time analysis with high sensitivity. On the other hand, this requires pre-sampling of the sample. So I'd like to introduce the method for sampling real sheet. There are passive sampling methods using canister and passive tube samplers, and active tube methods that use pumps to collect samples. As I mentioned above, there are various sampling methods, so it is important to choose the appropriate method based on the specific conditions of your experiment. Let me explain why I chose the passive sampling method. The experiment I conducted was a study that measured real sheet concentration at 25 air quality monitoring station in Seoul to understand the spatial distribution of real sheets. We measured real sheets over a long period across 3 seasons from 2022 to 2023 and analyzed the total of 52 real sheets, which are part of the EPA palm target compounds that could be analyzed using PAT. The square shown on the map represent Seoul's palms. As you can see, they are widely distributed. So, given the characteristics of the research, passive samplers were used because they are easy to install, cost-effective, and suitable for long-term monitoring across multiple locations. Passive sampling is especially useful for large-scale environmental monitoring or in areas with limited infrastructure, and is frequently used for long-term monitoring for research, or regulatory purpose. However, passive sampling only collects samples over a long period and provides average concentration, making it difficult to obtain immediate real-time data. Passive sampling offers long-term averages, but it may not capture short-term spikes or fluctuations. And the sample rate and measurement results can vary depending on environmental factors such as temperature, humidity, and wind. So if without proper adjustment for these external factors, errors can occur. Therefore, I compared and analyzed the real-time data from 4 monitoring sites in Seoul with passive sampling data. Next, I'd like to explain this in more detail. This picture shows a passive end sampler with the exposed part on the left and this diffusion cap used. The diffusion cap in the passive end sampler regulates the sampling speed, protects the sample from external environmental factors, and provides a suitable environment for long-term air quality monitoring. This ensures the accuracy of the collected samples and protects them from external contaminations, making it important for obtaining consistent data. The second picture shows the TDGCFID equipment used in the experiment. The samples collected were first analyzed in the TD, and the dissolved substances were analyzed in UDPC. We needed to analyze 52 times VOCs, so we used 2 columns and conducted the experiments with FID. And this table shows the analysis conditions of our equipment. Next, I'd like to talk about calibration. In GC systems, calibration is essential for ensuring the instrument can accurately measure specific concentrations of compounds. The calibration process uses standard gases at 1, 3, 5, and 10 ppb to establish a reliable correlation between the detector's responses and known concentrations. To ensure the precision of the experiment, gas standard analyses are performed after analyzing 7 samples for each concentration. These procedures involve routine checks following calibration to verify that the system constantly operates with the set calibration parameters, ensuring reliable performance over time. QAQC is also essential to ensure the reliability and accuracy of the data. We evaluate the precision with 5 different methods such as RMS, MAD, percentile, biases, and p-value of 23 VOC species from 72 duplicate sample sets. And this analysis provides important insight into the reliability and accuracy of VOC measurement using tests. Next section is peak alignment. Peak alignment in GC analysis is crucial for ensuring consistent comparison between samples, accurate data interpretation, and reliable statistical analysis. If the peaks are not correctly aligned, the same compound may appear at different positions, resulting the result and reducing the reliability of the data. To prevent this and ensure accurate analysis, peak alignment is an essential process. We have systematized peak alignment to accurately estimate concentration. Now I will discuss the content of my research, which studied VOC using passive sampling. This research aimed to understand the characteristics of pumped VOC emission in Seoul and evaluate the accuracy of VOC emission inventories by comparing measured VOC concentrations with TAPS model results. The spatial distributions of VOCs in Seoul area was obtained through past observation with consistently high concentration found in the southwestern region during all 3 observation periods. Its high concentration is believed to be due to the concentration of VOC sources such as heavy traffic, automobile paint shop, and laundries. Particularly, the western area of Seoul shows the high VOC level due to the cumulative effect of nearby business, the metropolitan landfill, and the airport. Regionally, higher VOC concentrations were recorded in central Seoul during the summer, likely due to increased evaporation of organic solvents in the hot weather. The following shows the spatial distribution of isoprene, a representative compound of natural emission, and the toluene represents the anthropogenic emissions. Isoprene showed high concentration in forest area, particularly on the outskirts of Seoul, reflecting the basin-shaped topography. On the other hand, anthropogenic VOCs like toluene associated with vehicle emissions and solvent use show higher concentrations in central and southwestern Seoul. To ensure data reliability, we compared VOC data from 5 active pump stations in Seoul with the past data. The graph shows the PVOC concentration measured at pumps and past observation sites. Significant signal variation were observed at urban pump sites, while no signal changes were detected in residential and background areas like Gwangjin-gu and Bukhan Mountain. Pasts recorded a higher concentration during the summer, but the results from pumps and pasts were statistically similar. Next, we performed PMF analysis using past data and compared it with actual emission data. CAPS is a comprehensive database platform that manages the emissions inventories of major air pollutants in Korea. The PMF analysis derived area sources showed high VOC concentration in southern and southwestern Seoul, consistent with CAPS data. However, unlike the CAPS data, PAPS did not identify major emission sources in southern Eastern Seoul, likely because the large automobile painting shops were more than 5 kilometers away from the nearest PAPS site. This shows the special limitations of passive sampling. In the road source section, the spatial distribution of road traffic emissions derived from the PMF analysis largely aligned with the CAPS data, showing the elevated concentration in central and southern parts of the Seoul. However, in Eastern Seoul, PAPS sites were relatively far from major roads, resulting in discrepancy. Therefore, caution is needed when directly comparing PMF results with CAPS data. And we also analyzed 52 VOCs and grouped them into 8 categories before comparing them with CAPS data. First, let's discuss the point of agreement in the VOC compositions. In both the PAPS and CAPS data, aromatic components make up more than 50% of the PVOCs. This highlights the significant contribution of aromatic to overall VOC level in Seoul. Among the major components, we see that aromatic 1, alkane 2, and 3 are the dominant species in both datasets. These compounds are responsible for a large portion of the total VOCs concentration, indicating consistency between the two methods in identifying the key VOC contributors. Now let's move on to the discrepancies between the two datasets. In the past data, the most dominant species was aromatic 1. However, in the CAPS inventories, aromatic 2 was the most dominant. This suggests a difference in the identifications of the key aromatic compounds between the two methods. Another key difference is in the benzene level. In PAPS measurement, benzene accounts for 4% of the total VOCs during the winter, whereas CAPS consistently reported benzene level at 12% throughout the year. This indicates that CAPS may might be overestimating the benzene concentration or not capturing signal variations accurately. Lastly, we modeled VOC concentrations using both the box model and Gaussian flow model, comparing the emission rates with actual measurements. The box model requires VOC's emission rates and wind speed, boundary length, and PBL height and background concentration, while the Gaussian model uses the vertical dispersion parameters based on atmospheric stability. VOC emission rates were calculated using the 2021 CAPS inventory, and background concentrations were obtained from Bhutan Mountain with meteorological data from the Otan station. Both the box model and Gaussian model show similar VOC concentrations ranging from 12 to 15 ppb, with more pronounced seasonal variation in the box model. The lack of detection of emission hotspot in southern eastern Seoul in the past data, coupled with the omission of photochemical loss in our dispersion, only model indicates that the current CAP emissions inventory may significantly underestimate actual ambient VOC levels, especially in summer. As a result, future research should include more precise spatial observations and photochemical reactions, as CAPS may not fully capture all VOC emission sources. So in conclusion, I assess VOCs in Seoul using both PATS and actual sampling from 5 pump stations, confirming that PATS results align well with online pump data in capturing spatial temporal VOC behavior. Spatial analysis using PATS highlighted localized and transported VOC sources in southern western Seoul and vehicular emission in the urban center, consistent with CAPS data. CMF analysis identified general agreement between PATS and CAPS data, though discrepancies were noted in southern eastern Seoul, emphasizing the need for denser PATS observation. Plant CAPS emission inventory may significantly underestimate actual ambient VOC levels, especially in summer. Let's conclude in the discussion about my paper. And finally, to summarize today's lecture, there are various methods for measuring and analyzing VOCs, including GCFIDs, MES, PCR-MES, canister sampling, and passive and active tube samplers. Among these, passive sampling is a cost-effective method suitable for long-term monitoring, and it's particularly effective for assessing spatial distributions over large areas. However, since it cannot capture short-term fluctuations, it is important to compare passive sampling data with real-time sampling data. In VOC analysis using GC with passive samplings, regular calibration and QA/QC procedures are essential to ensure data accuracy. Peak alignment in GC analysis is also crucial for guaranteeing consistent results, and I have systematically applied this to ensure accurate concentration measures. If these aspects are carefully considered, passive samplings can be regarded as one of the accurate and effective methods for VOC concentration estimation. That's all for today's presentation. Thank you for listening. I hope this lecture was helpful to you all. Thank you.